What is an ERA and why does it matter? An environmental risk assessment, or ERA, evaluates the potential environmental impact of human medicinal products. It assesses whether and to what extent active substances or their metabolites may enter the environment after use, for example via excretion or improper disposal, and subsequently reach surface waters or soils. Some substances are persistent, bioaccumulative, or toxic. Over time, they can cause long-term effects on aquatic and terrestrial ecosystems. The purpose of an ERA is to identify these risks early, before they turn into environmental or even public health concerns. If environmental risks are identified, the assessment must also propose appropriate risk mitigation measures. These may include, for example, specific warnings or instructions in the product information. Who needs an ERA? In short, all applicants for a new marketing authorization for human medicinal products in the EU. This applies to new active substance and generics alike, and to all authorization procedures. Since the revised EMA guideline entered into force in September 2024, there are no exemptions. An ERA is also required for Type 2 variations and extensions for marketing authorizations. In addition, the planned reform of EU pharmaceutical legislation is expected to significantly increase requirements, introducing mandatory risk mitigation measures and allowing for post-authorization data requests. Regular review of ERA data is also foreseen. Where necessary, the ERA must be updated. What does an ERA actually include? The assessment follows a stepwise approach. Phase 1. The potential environmental risk of the active substance is evaluated. The predicted environmental concentration, PEC, in surface water is calculated. If the PEC is greater or equal 0.01 microgram per liter, a Phase 2 assessment is required. Phase 2 studies on ecotoxicity, degradation and distribution in surface water, groundwater, soil or sewage treatment plants. In addition, APTB assessment determining whether the substance is persistent, bioaccumulative and toxic. The outcome is clear: either no environmental risk or potential risk, and, if applicable, concrete measures, for example, relating to use or disposal. Conclusion: the ERA is no longer a bureaucratic add-on. It has become a core element of sustainable pharmaceutical development. Those who address it early save time, costs, and regulatory discussions later on.